GAVIN HUNTER, and I'm a researcher from the CSIRO based in Canberra. I'm a plant pathologist that works in the CSIRO temperate weeds team, where we develop classical biocontrol solutions to invasive weeds in Australia. Today I'll talk about the deployment of the first biocontrol agent for sea spurge across southeast Australia. This work is done together with several of my colleagues, including Caroline Delas, Isabel Zale-Rolfe, and Louise Morin. I'd like to begin by acknowledging the traditional custodians of the land on which we meet today and pay my respect to their elders past and present. I extend that respect to all Aboriginal and Torres Strait Islander peoples here today. In this talk, I'll provide information on sea spurge, its native range and its distribution in Australia, the history of biocontrol research into sea spurge, the surveys that were undertaken and where the biocontrol agent was collected. I'll detail the host-specificity experiments that were undertaken in Australia with the biocontrol agent and talk about the infection process employed by the agent on sea spurge and what some of the characteristic disease symptoms are. I'll then briefly discuss the process whereby the application to release the agent was submitted to the Australian regulator and talk about some of the initial work that we've done this year in developing and testing methodologies to release the biocontrol agent in the field. And lastly, I'll focus on the current project, which is the wide-scale release of the agent in Tasmania and Venturia, and what realistic expectations we should have from this project. So sea spurge is native to Northern Africa, Southern Europe, and Western Asia. It's widespread along the coastline of countries along the Atlantic province and into the Mediterranean. In Australia, sea spurge is invasive along the southern coastline, infesting dune ecosystems from just above Perth in Western Australia all the way around the southern coastline to the mid-north coast in New South Wales, and it's known from all bioregions in Tasmania. Sea spurge was first collected in Australia in 1927 at Albany in Western Australia, and then again in 1937 at Port Victoria in South Australia. It's believed that the plant was likely introduced into Australian waters by the ballast water of ships travelling from Europe to Australia. The plant is a perennial herb with 1 to 10 stems and can produce thousands of seeds per plant. Following seed production, stems usually die and are replaced by new shoots from the root crown. And long-distance dispersal is by seed, transported by ocean currents, or by accidental anthropogenic movement. The CSIRO, with funding from the then Tasmanian Department of Primary Industries and Water and the Australian Government through its Rural Industries Research and Development Corporation, undertook surveys in France and Spain for any pathogens and herbivorous arthropods of sea spurge during 2008 to 2009 to try and find possible candidates for classical biocontrol of sea spurge in Australia. 22 sites, sea spurge sites along the Mediterranean and Atlantic coast of France were sampled, and these collections resulted in the collection of 4 insect species and 2 fungal species, one of which, called Venturia paralias, produced these impressive symptoms on sea spurge plants. After some initial testing with this fungus at the CSIRO European Laboratory in Montpellier, it was decided that this fungus was a promising candidate for further, more comprehensive host specificity testing in Australia. So in order to carry out host specificity testing for candidate biocontrol agents, we had to develop a comprehensive host test list of non-target species. This is accomplished by using a technique known as the centrifugal phylogenetic method that is based on the phylogenetic relationships of test plants to the target weed. This method is underpinned by evidence that specialist candidate biocontrol agents are evolutionarily more likely to infect non-target species that are closely related to the target weed relative to those that are more distantly related. So in terms of sea spurge, we investigated published phylogenetic and taxonomic literature from the family Euphorbia to develop a list of related Euphorbia species to include in host testing experiments with the candidate biocontrol agent. The final test list included 44 non-target species from the family Euphorbiaceae, Picridendraceae, and Phyllobaceae. Sea spurge is accommodated in the subgenus Eschula within the genus Euphorbia, and we managed to include 16 non-target species from subgenus Eschula in the host testing and several from subgenera Aphthymalus, Chamaesis, and Euphorbia. In 2017, the New South Wales Government, through the New South Wales Environmental Trust, provided research funding for CSIRO to undertake comprehensive host specificity testing with the fungus Venturia paralias on sea spurge. This funding has allowed us to develop mass culturing and release techniques for the fungus. The fungus was imported into the biocontainment facility at CSIRO in Canberra, where we proceeded to test if the fungal agent was specific to sea spurge. Essentially, we would mass culture the fungus in growth chambers and then harvest the fungal spores to produce spore suspensions, which were sprayed onto non-target Euphorbia species from the host test list and onto sea spurge plants that acted as positive controls. Following inoculation, the plants were assessed over a 30-day period to see if any signs or symptoms of infection occurred. We would also take leaves from inoculated plants at 2, 7, and 12 days post-inoculation, and these were viewed with light microscopy to see if and how the fungus infected the plant species. This is a table indicating some of the results from the host specificity testing and the microscopy observations. You'll see the furthest column on the right-hand side specifies the plant response to the biocontrol agent. For this, we specified 4 response categories, namely highly susceptible, immune, and resistant. These were based on the visible symptoms observed on the plant and microscopic development stages of the fungus as defined by 17 fungal development steps on and inside the plant. The take-home message here is that sea spurge is highly susceptible to the fungus, and the fungus was able to penetrate sea spurge leaves, grow within the plant, produce reproductive structures and spores, and complete its life cycle on sea spurge plants. You'll notice that all the other species except Euphorbia digitalis were either characterized as immune or resistant to the fungus, meaning that the fungus was not able to penetrate and grow inside the plant, and the resistance of the plant to the fungus kicked in at that level. For Euphorbia digitalis, this species was characterized as susceptible to the fungus. And this was because of the fungus was observed to cause small, restricted leaf lesions on a few leaves of 2 out of 10 plants inoculated with the fungus, and these plants did not die from the infection. It's important to note that Euphorbia digitalis is an exotic plant native to the Mediterranean. It's a very close congener of Euphorbia paralias and is regarded as a naturalized weed in Australia with only a very limited distribution in the environment, if any. So in summary, of the 44 non-target species that we tested, all of them except Euphorbia digitalis were either resistant or immune to the candidate biocontrol agent of sea spurge. During host specificity testing, we were also able to determine the method the fungal agent uses to infect and proliferate within sea spurge. Once fungal spores land on sea spurge leaves, they germinate and produce infection structures called aprosoria. The fungus penetrates the leaf cuticle and grows in the leaf cuticular space, producing primary amoeboid hyphae, and then these stolon-like hyphae. By 12 days after inoculation, a well-developed network of subcuticular hyphae are visible, and the leaf lesions are usually observed under laboratory conditions between 11 to 14 days after inoculation. Eventually, these camelliaphores that are the fungal sporulating structures are produced from which the fungal spores are formed. In terms of the symptoms, once the lesions are seen on the sea spurge leaves, the fungus is able to move from the leaves into the stem of the plant, where it causes these characteristic dark brown to black stem lesions that extend up and down the stem. Eventually, these lesions hinder the movement of water and photosynthase through the plant, and in severe infections, the stem loses turgor and can drop or fold over. So once the host specificity testing with the biocontrol agent of sea spurge was completed, the data and results were compiled and an application to release the biocontrol agent into the Australian environment was submitted to the Australian regulator, who then proceeded to undertake a comprehensive risk assessment of the information, data and results that were presented in the application. Once the regulator had produced a draft risk analysis, it was submitted to the Plant Health Committee for their feedback and also underwent a public consultation. After this whole process, we received notification in November 2020 that the regulator recommended the release of the fungus for the biocontrol of sea spurge, and that the overall risk associated with the release was negligible and that it achieves Australia's appropriate level of protection. Once we obtained permission to release the sea spurge biocontrol agent, it was removed from the CSIRO biocontainment facility to our laboratories, where we set about developing and testing various methodologies that could be used in the field to inoculate sea spurge and release the agent. We were very fortunate that Kerry Thompson, the Natural Areas Operations Officer for the Shoalhaven City Council in New South Wales, kindly suggested 2 sites where we could trial the initial field releases. So in April this year, we released the fungus at Cadbury Beach and Conjurum Point in New South Wales using a few different methods, including preparing spore suspensions from dried culture material of the fungus and spraying this onto sea spurge plants, doing the same thing but using fresh cultures of the fungus, using sea spurge stems already infected with the fungus and tying these to the healthy sea spurge stems, or using whole sea spurge plants infected with the fungus and planting these alongside healthy sea spurge stems. We've subsequently been back to these sites on 2 occasions since we did the initial release and inoculation of sea spurge plants. At both Cadbury Beach and Conjurum Point, we've started to see symptoms on the inoculated sea spurge plants caused by the biocontrol agent. More specifically, these characteristic dark brown to black stem lesions that you can see in the pictures. Most importantly, we'll soon be ready to move into the next phase of sea spurge biocontrol using the biocontrol agent. And this is the community-led releases of this agent in Tasmania and Victoria. Once again, the New South Wales Environmental Trust has provided research funding for a 3-year project that started this July and will run until June 2024. We know that severe sea spurge infestations occur in Tasmania and Victoria, and these infestations are likely the source of seeds that spread via ocean currents and invade beaches in New South Wales. So, to help stop the spread of sea spurge, this project will help to support releases of the biocontrol agent in Tasmania and Victoria. Ideally, with the help of community members, we'd like to have releases of the agent done at many coastal locations. And if you or your organisation would like to participate in making releases of the agent at sea spurge infestations in your vicinity, we would really like to hear from you. Feel free to contact me and we'll get you registered and your details into our database so that we can send you dried material of the biocontrol agent and detailed guidelines on how to make field releases of the agent. It's also important to think about what we should expect with this biocontrol agent of sea spurge. This agent has never been released anywhere else, so this will be its first deployment outside of its native range to help manage sea spurge. So we don't yet know if the agent will survive and persist in Australia, but we'll be able to determine through this current project. Testing undertaken in the laboratory is always under ideal optimum conditions, and we were able to define the biology and symptomatology of the fungal agent on sea spurge under conditions that are most suitable for its growth. However, in the environment, the establishment of the agent will be determined by the prevailing environmental conditions. In order for the agent to have the greatest chance of establishing in Australia, we believe that many field releases of the agent should occur over as wide a range as possible, so that the agent can cause severe disease symptoms on sea spurge year after year. Biocontrol of sea spurge is not a silver bullet. It's a long-term strategy that will depend on the agent becoming established, and once it does, it will form an important part of an integrated management strategy, employing existing sea spurge management strategies and the biocontrol agent. So in the end, I think there has to be some management of expectations, and we'll have to wait and see until the end of this project to get a good indication as to whether the fungus is a good biocontrol agent or not. And finally, I'd just like to acknowledge the following organisations and people for their help and support. The New South Wales Government through the New South Wales Environmental Trust for ongoing financial support to undertake research on this biocontrol agent, to our many collaborators on this project who have been a great help to us, including Kerry Thompson from the Shoalhaven City Council in New South Wales. In Tasmania, we thank John Marsden-Smedley from Sprats, David Pemberton from the Tasmanian Parks and Wildlife Services, and Peter Stronach from Landcare Tasmania. In Victoria, I'd like to thank Parks Victoria, especially Mark Rodriguez and his many colleagues who have been very helpful and eager to help us identify monitoring sites and who would like to participate in the project. And most of all, the many friends groups and community organisations in Victoria and Tasmania who are keen to partner with us to make the releases of the biocontrol agent across Southeast Australia. Please feel free to get in touch with myself or my colleague Caroline if you have any questions or if you're interested in making community-led releases of the biocontrol agent. I would be very happy to answer any questions that you may have. Many thanks for all of your time and attention. Thank you.